I'd like to ask Ian Gibson, one of our lifetime members and awardees, to please introduce our speaker of the night, Danny Miller.
Okay, everybody here? It's a pleasure for me to introduce my good friend, DNA Miller. He has joined me for the last half of the MatchMaker MycoMatch journey, and we've exchanged thousands of emails on that. We're both active participants in the Pacific Northwest Key Council. In Puget Sound Mycological Society, DNA has several leadership roles, and he's at present one of the most active people in collecting DNA sequences from Pacific Northwest fungi. He's a Canadian. He grew up on the Niagara Escarpment in Ontario, and he likes to go back there for family visits, of course, usually in the summer, but occasionally he likes Niagara Falls. His university years were at the University of Waterloo, which I assume is where they invented the flush toilet. After the university, that Microsoft nabbed him. They heard that he was good at breaking windows, and he accepted and moved to Seattle on one condition, that they would allow him to ski every month for the next 20 years. And that's what happened. So without further ado, DNA Miller spoke to us in 2017 to South Vancouver Island Mycological Society, but a lot has happened since then, and I'll turn it over to him now.
Thank you. It's great to see everybody again, even though it's only virtual, and I'm really looking forward to the time when I can come again in person. That'll be great. Tonight, I am going to talk about the whole process of DNA and DNA sequencing and what it means for mycology and how it works. It's kind of the biggest revolution since the invention of the microscope, and also give a little bit of insight on kind of what a day in the life of the modern mycologist is for those who are looking at doing the DNA work. So the first thing I want to say is that as humans, we love to classify things. Just knowing the names of thousands of species or everything that's out there in the world isn't enough. We kind of want to make diagrams like this tree here to kind of explain and guess of how these different organisms might be related to each other, and kind of make a tree of which ones are related, which ones aren't, and sort of, we just love to organize and classify things. And in the beginning, what we could do without a microscope, without anything, this is back in the 1700s, everything started by classifying them basically by how they look to the naked eye without being able to get too close a look at it. So we used to assume that everything that was similar and that everything that looked the same as another mushroom must be closely related to it. So Elias Fries, who's sort of the father of mycology, in his books that started in 1821, began to classify all the mushrooms, and he had a few different types. Agaricus were all the ones with gills, and Boletus were all the ones with pores, like the porcini. And he was the person who decided how to classify them and what features probably meant that they really were more closely related to each other. So the first thing that he did was whether or not underneath the cap you have gills or pores. And the second thing he did for all of the gilled mushrooms is say that the second most important thing is going to be the color of the spores. So for all of us who hate the fact that whenever we get a mushroom, we have to wait a few hours and make a spore print by taking it off, putting it on the paper, and seeing what color spores there are. You can blame Elias Fries from back in 1821 for being responsible for making all the keys in the books dependent on knowing that feature. And well, you can't really blame him because it turns out that is a very important thing. If we had kept Agaricus as the genus for all gilled mushrooms, there would be 10, 20, 30, 40,000 mushrooms in that one genus. So it has since been split into many, many, many different genera. But it's just far too unwieldy to contain them all. So it is important that we look at things like spore print and things like that. But he's the one who first suggested that back in 1821. And some tough decisions had to be made. Mushrooms don't always cooperate. They don't always fall neatly into one category versus the other. There's some things that are in between. For instance, he described Agaricus as all the mushrooms that had gills under the cap, but also noted that they were all often very soft and not tough to chew. You could actually eat them without breaking your teeth on them. So they had gills, they were soft, and they often grew on the ground. And on the right-hand side here, we have the polypores that he described. When you look under the cap, you see a kind of like a sponge, a bunch of little holes, and they're very hard and tough, and they often grow on wood. So then along came this mushroom in the middle. It is a hard, tough conch growing on wood, but when you turn it upside down, it had gills. So which is it? It's kind of half one, half the other. And he had to make a decision. So the decision he made was, as I said in the last slide, the number one important thing is what's under that cap, gills or pores. So he called it an Agaricus, and he called this a gilled mushroom. And that was what the rule said. But nobody really knew for sure if that was the right thing to do. So then we invented the microscope, and that kind of shook things up. When we looked underneath under the microscope at the gills where the spores were growing and looked at the spores under the microscope, we almost always saw, for gilled mushrooms, these ellipses about 10 microns by 6 microns would be a sort of an average size. And all of the other tissues besides those elliptical spores that you can kind of see in the background are these very long, stringy fibers. And that's what you normally saw. And then they put a Russula under the microscope, and it looked completely different. The spores were these perfectly round things, and if you put iodine on them, they would turn black warts would appear. They're not black, but they're covered in these black pimples that you see when you put them under iodine. Very different. And not only were the spores different, but all of the other tissues in the background that's kind of blurry are not long and stringy things. They're other mostly circular things. There's just circles all over the place in the microscope for the Russula. So that was when we realized that things that were invisibly small to the naked eye might actually turn out to be much more important for discovering what something really was than any obvious difference that you could just see with the naked eye. For instance, boletes, those soft, spongy pored mushrooms like the porcini, all pretty much had the same kind of spore. They were 2 to 3 times longer than they were wide. So they weren't really ellipses, they were much more elongated than that. And then when they came into the forest and they found this mushroom here, it looks an awful lot from the top like a few of our boletes, with that kind of a brown felty cap and the whole shape and stature of it. And you expect it's a bolete, then you pick it up and you turn it upside down. And as you can see, there's bright yellow gills under the cap and not pores. And even the fact that they're bright yellow, boletes are, many different boletes that look like that are bright yellow underneath when you turn them upside down. So the color matched, everything matched, except there were gills where there were supposed to be pores. And then they threw it under the microscope, and lo and behold, it had the same spores as the boletes did. So this couldn't be coincidence. This was the sort of first clue that things were not being accurately classified when we said, if you have gills underneath the cap, that's the #1 indicator of how you might have actually evolved. And this sort of gilled bolete is a close relative of some of the boletes. And then you have the gilled mushrooms, and you have a Russula, which is a third thing altogether. It's its own kind of mushroom with its own branch of the family tree. That's unrelated, that is not closely related to either of them. So those were some of the really cool things that we learned through the invention of the microscope. So therefore, every mushroom was eventually classified, and they relaxed the rules a bit. If the microscope showed that these similar looking mushrooms were actually probably very different, or vice versa. They were allowed to classify the gill bolete as a kind of bolete. But did they get it right? And a really good question, is there really even any such thing as a right answer? Because I might think of that hard conch that grows on trees with the gills underneath as a gilled mushroom that just happens to be tough in texture and grow on wood. But another person might think of that conch as a hard conch growing on wood that just happens to have gills when you turn it upside down. So who's right? Is there a right answer? Is it all just really a matter of opinion? And then DNA came up. And we've kind of all collectively made an official decision that if you are genetically related to somebody, then we will say that you are related to each other. This actual genetic family relationship is the true kind of relationship that matters. So in other words, if two mushrooms had the same great-great-great-great-great-great-great-great-grandparent, that means that they were closely related, no matter what they look like, no matter anything else. They are closely related in the same kind of family. And this definition is probably the right one and has the most meaning, because if you have a bunch of mushrooms that are deadly poisonous and you're looking to find more deadly poisonous mushrooms, you're much more likely to find one that is a cousin species to the other one. It is much more likely to share those genes that make it deadly poisonous than some randomly unrelated mushroom that just happens to look similar. So that really probably is the best way to classify things, to learn things about which ones are going to be toxic, which things they would also share ecological and habitat behaviors. So if one of them is going to grow in a certain kind of environment and be threatened by habitat loss, this related one might be subject to the same risks. So this is a very useful way to think about how we classify the mushrooms. And it is now how we are going to classify them. So what is DNA? Well, it's a very long string of molecules that you can represent as a long string of letters. So for instance, it's this double helix that I'm sure you've heard of, on here. And there's just all the sort of ladder rungs are 4 different possible molecules that could be on the ladder. There's the red molecule, thymine, which we call T. There's the blue molecule, C. The yellow one is A and the green one is G. So if we were to start at the top right-hand corner here and wind our way down in the direction of the arrow, and we were to read what colored molecules, we would see that it's blue, then yellow, then green, then yellow, then blue, then red, and on and on and on. We could, of course, read it from upside down, going up. And if we start on the bottom left, the molecule colors are blue and red and green and yellow, and etc. So when we read the DNA strand, we simply say the letter name of the molecule, like A, C, G, T, C, C, G, T, as we're reading up. So reading from the top down of blue, yellow, green, I would read C, because that's the blue, A, because that is the yellow, and then G for the green one. So this information sort of encodes almost everything you need to know about an organism. And it takes a lot of data. Of course, our DNA strands are about 3 billion long in humans. So 3 billion of these numbers, it's quite long. Mushrooms are not quite as complicated as people. It's only about 50 million or so DNA characters make up the description of a mushroom. And now you probably know that we have 23 chromosomes when we talk about our DNA. What that means is all 3 billion characters are not in one giant DNA string. It's broken up into 23 pieces, each one called a chromosome. And if you count all of the molecules on all 23 strands, the total will be about 3 billion. So the sort of biological rules, if you take a look at this strand, is that blue always connects to green. In other words, C and G go together. And red always connects to yellow. T and A go together. So you'll notice in this diagram, blue and green are together, red and yellow, blue and green, red and yellow, and that's always the way it works. So we, reading it forwards from the top down, starting at the 5 end, they call it the 5 end for some reason, you can read the DNA strand. It's blue for C, yellow for A, green for G, etc., all the way down. And there you see I've printed it, whoops, right here after the 5 prime, C-A-G-A-C-T, blah, blah, blah. And if you read it upside down from the 3 end, you get blue for C, red for T, etc., and you get the strand backwards. So if you know one of the directions, you can easily figure out what the other direction is, because it's just the same letters, but written backwards. And you change all of the letters to their opposites. So if you take a look at these 2 strings of letters I've got here, there's a C on top of a G and an A on top of a T, because C always goes with G, A always goes with T. And so there's 2 ways to read the strand, and we've sort of decided that reading it from the top down is going to be the forward way that we're all going to read it so that we can all talk about the same thing. And this sort of relationship where it's reading reading from the 2 different directions gives you the string backwards with all of the letters switched is called anti-parallel. So the DNA strand is anti-parallel. Now, there's a really interesting parallel between DNA and computer programs and computer hardware. It's almost like living things are a kind of computer. It's kind of it's almost scary the way this works out. A DNA sequence is just like a computer file, like a picture that you have on a thumb drive, your favorite JPEG of your vacation pictures. That's pretty much a great analogy for what DNA is, because if you have a GIF file on your thumb drive, and you can tell because it actually ends in the extension GIF, but if you actually look at the file, the letters GIF are the first 3 letters in the file, and that's how it tells the computer that this is a GIF file that it should print on the screen instead of being an Excel spreadsheet, for instance. When you have an Excel spreadsheet file, if you were to look in that file, every Excel spreadsheet file starts with D0CF11E0, blah, blah, blah. And that is the key to tell the computer that it is an Excel file. So when the computer sees that, it knows what to do. It knows to display that data as a spreadsheet and not try and draw it as a picture. So for instance, the gene in your body that indicates what your eye color is going to be might start with this certain string of letters. And that's its code. And that tells your body whenever you see these letters, what's coming next is the eye color. And that's how your body reads your DNA to figure out what to do. So humans, we count in base 10. We have 10 fingers. So we decided to count in base 10, where we count from 1 through 10, and then we start all over again with 11, 12, 13, 14, 15, 20 again. Computers, as you probably know, use base 2, binary. They just use the numbers 0 and 1, and they're happy going no farther than 0 and 1 in a single digit before having to go on to a 2-digit number. So it turns out biological organisms use base 4 because there's 4 possible digits, A, C, G, and T. So, so DNA and biological organisms basically count in base 4. And all software needs hardware. If you had a thumb drive with your pictures on it, you can't just hand the thumb drive to somebody and say, hey, look at these beautiful mountain pictures that I took. There's no way to see what's on there. You need a computer, you need a piece of hardware to be able to read that picture software and draw the picture to show it to you. And in the same way, DNA by itself is useless. You need your body, which is the hardware that will go in and look at that DNA, which is the software, and figure out what to do. Make your eyes green, make your hair red, in my case. So we, as humans and mushrooms, are what's called diploid organisms, which means we don't just have a very long strand of DNA that describes what we are. We have 2 complete sets of DNA, a complete description of basically 2 separate people inside of us. So which of the 2 DNA strands is the real us? Well, neither one by itself the real you is a combination of the two. So for instance, if person number one's DNA that's inside of you says you're to have brown hair, and person number two is a redheaded person, you will have brown hair because brown hair is the dominant trait in that particular case. So there's sort of an interaction between the two descriptions that describe you. And for simplicity, I'm not going to always show the DNA strand as the double helix with the two copies of everything. I'm showing one strand of each helix here on the left. So basically, the two copies of DNA that are inside you are very close to each other. They're not radically different. In this example that I've shown here, there's only two places that they're different. I've marked them with an X. Instead of an A on the left, the second copy of DNA inside you has a T. And then it's mostly the same. And then when you get down here, the first copy of you has a C and the second one has an A. But they're mostly the same. And just one fun fact is that if you are a genetic male, one of the two people inside you is a genetic female. So I have one quick slide about how sexual reproduction works because I think it's interesting. And the way it works is your body knows which sections of DNA are genes that perform a function and where it changes to a different gene that does something different. And what it does is your body will take a random copy of a particular gene, either the left copy or the right copy, depending on randomness, and put that into your sperm or your egg and create a new strand on the right, which is the left-hand side for whether you have brown or red hair, for instance, the right-hand side gene from whether or not you're right or left-handed, right-hand side gene for whether you're a boy or a girl, the left-hand side gene for whether you're going to love or hate cilantro, and it puts those random segments together to make a new single strand, which is kind of a mixture of the two strands inside you. And then when the sperm and the egg meet, you get one strand from each parent, and you go back to having two strands that can completely describe a person, and they interact to make your child. That's kind of interesting. So we want to DNA sequence a mushroom. How do we do that? Well, 50 million base pairs is quite complex and long, and we don't need to do it all. That would be pretty unwieldy and still very, very expensive. And especially in order to interpret the results, you kind of have to read all of the characters by hand. So if you had to actually look at all 50 million, you'd maybe be able to look at one DNA sequence every year, and that was it. That would be very unwieldy. So typically, we only want to sequence a very small part of that, less than 1,000 characters. That's plenty. And it turns out there's a convenient spot of the DNA called ITS, the internal transcribed spacer, and it's kind of something we call junk DNA, which means it doesn't really do anything. It's there, and your body is never going to look at it. It's never going to look at that part of your DNA, so it doesn't matter what's there. It's free to mutate and change as much as it likes without mutating in a way that could kill the organism, and therefore that gene will never be passed on. So it's about 600 to 700 pairs long. It's a nice size, nice and short. And given that it's a junk piece of DNA that mutates quickly, you will see differences between two species show up pretty quickly. Two different species will usually have different ITS DNA. So it's a really good thing to check to see what species you might have. And only mushrooms have this part of DNA, this ITS junk region. So if you're in a lab and you're reproducing DNA and you touch something, you get your own DNA on it and contaminate it, it used to be a problem. You had to do DNA sequencing in a very sterile lab and hold your breath and wear gloves, because if you touch something, your DNA would get on it and you'd accidentally sequence your DNA and not the mushroom. So the handy thing is, this is a part of DNA that humans don't have. So when you put the chemicals in to sequence this particular part of the DNA, it will ignore human DNA And as long as you don't have more than one mushroom contaminating what you're working on, you don't have to worry about you yourself contaminating. So that's one kind of method that sequencing things has become much easier than it used to be. So as I said, this ITS region is really great to see if two mushrooms belong to the same species. If you want to know if they're in the same genus or the same family and figure out relationships that are further apart, there are other different genes that mutate more slowly that are ideal for answering those kinds of questions. So depending on what you want to do with your sequence, it depends which part you're going to sequence. So let's talk about exactly how DNA sequencing works, because I think it's pretty interesting. It comes in two parts. First step is called PCR, polymerase chain reaction. And what it's going to do, it is going to make millions and millions and millions of copies of just the tiny piece of DNA that you wanted. So you'll have a billion copies of that little 700 length strand that you're interested in by the end of this process. And like every recipe, it has ingredients. Here's the ingredients we need to do our DNA replicating. You need a piece of the mushroom. You need a forward primer. That means you need to know what the beginning of your region of interest looks like, what letters show up at the beginning of the piece of DNA you're interested in, so it can find the piece that you're interested in that very long string of 50 million. And you also have a reverse primer, which means you have to know the letters that come at the very end of the region that you're interested in. And it's going to sequence everything in between the beginning and the end. And that's how it's going to know to get just the piece you're interested in. You need an enzyme, which triggers DNA copying. So we need one of those. And the last ingredient in our little recipe here is we need a whole bunch of spare individual molecules of A, C, G, and T just floating around in the soup. A whole bunch of spare single letter molecules floating around because we need a bunch of extras. So here we are. Here's a picture that kind of shows the soup that has all our ingredients in it. At the top, we have a bunch of loose molecules of all the different letters. We have our enzyme, which does DNA copying, which we'll talk about later. It looks something like that, I imagine, or that's a clever artist's rendition of what it looks like. Here in the bottom is our DNA strand we're interested in. And in between the two bars is the small piece that we're interested in copying. So we have the forward primer, which attaches to the letters, which our forward primer is CAG, and C always goes with G, A always goes with T, G always goes with C. So the CAG will find the GTC, and the GTC is always at the beginning, say, of the ITS segment. So we need to find it. So we have the letters that complement it. And at the end, we also need the letters that go on the bottom half of the double helix to mark the end of the region we're interested in. And that's all we need. So step one, you take the soup and you heat it up to 95 degrees Celsius, because at 95 degrees Celsius, DNA strands start to come apart. They actually unravel like a zipper. So the double strand where the G's and the C's and T's are always connected to A's, as you can see here, will start to open up, and you'll get the two halves separated. Then you drop the temperature to 55 degrees Celsius, because at that temperature, that's when the primers will find their matching numbers and snap into the right place and mark the beginning and the end of the part of the DNA that you're interested in. Now, for the sake of this example, I've used some pretty short, just 3 characters long, and you're probably thinking, there's got to be more than one GTC somewhere in the 50 million character long DNA. How does it find the right one? And you're right. It's actually 20 base pairs long. It's actually 20 characters long. My little diagram is only showing it as if it was 3, so that the diagrams are a little easier to follow. but the primers are actually about 20 long, and it is true that the only time you will find those 20 letters together in a DNA strand is at the beginning of the ITS region, because that's how your body works. It uses these unique keys to find the pieces of the DNA, and it has to work or nothing works. So I'm just showing them 3 long here to give them a little simplicity. So, okay, that's step 2. We've got the primers in place, and one more step, step 3. You heat it back to 72 degrees Celsius, and this particular enzyme is an enzyme that allows DNA to replicate at 72 degrees Celsius. That's the temperature that this enzyme works at. And then if that enzyme is there, and if your DNA is at 72 degrees Celsius, what happens is all of the loose letters that were floating in your recipe will find their places and snap into place one at a time. They'll attach at the end. This T, because it's an A, only a T will fit there. This G will find it's made of a C. The A will snap in, the C, the T, the A, and they'll go click, 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 and they will duplicate the DNA and put the second half back. So now, instead of having one piece of DNA, you have 2 identical pieces, and you've copied it. Only you haven't copied the whole thing. You copied starting at the beginning and moving forward. You didn't copy the stuff you weren't interested in that was earlier on. And for the second half here, you started here, and all the characters will snap in place and go to the left, and you are not copying the part at the end of the DNA which you didn't care about. So the speed at which these molecules find and snap into place is about 1,000 of them every minute. And we are only sequencing about 600 or 700 characters of DNA. So 1,000 is more than enough. A minute is more than enough. So we keep the temperature at 72 degrees Celsius for 1 minute, and we can be pretty confident that it has copied our whole ITS region for us. And that's it. We've duplicated the DNA, and now instead of 1 copy, we have 2 copies. So now we just repeat this 30 times. We go back to 95 degrees, and both strands will split. We go back to 55 degrees, and the primers will click in place again on both of them. And then we go back to 72 degrees for another minute, and then the 2 will now become 4. And we do this 30 times. And by the time we've doubled 30 times from 2, 4, 8, 16, 32, it goes up pretty quickly. There will be more than 1 billion copies of the piece of DNA that you want after doing it 30 times. And how long does it take? Well, I already mentioned that you have to leave it at 72 degrees for about a minute. Nothing else needs to be left on for that long, just a matter of seconds. So a total of 30 minutes to do this 30 times, maybe 40, 45 minutes, be sure, is all the time it takes to do this step 1 of DNA sequencing, the first half, which is the PCR to make all the copies. And there you go. So as I say, after you've done it twice, you'll have 4 copies. And look what's happening. The very long one, it only, it left off the beginning the first time. And the second time, it's going to leave off the end. Most of the 1 billion copies are just the tiny piece of the exact size you wanted them. There's only a few longer ones left around. Almost every one of the billion copies is just that 600 or 700 characters that you wanted. So that's great. We have 1 billion copies of the DNA. Now I want to stop and say something about this enzyme, which is pretty cool. Back in the old days when they were just figuring out DNA, one of the problems I already mentioned was that you couldn't touch anything, or you'd get your own DNA on it and you copy your own DNA by accident. So by finding parts of the DNA that don't exist in humans, you don't have to worry about touching things anymore. But a second really annoying thing about copying DNA was that you had to go stand in a deep freezer in order to do it, because all of the enzymes we knew of would duplicate DNA at room temperature, and it would always be duplicating the DNA, and you have to turn it off. It has to only be in step 3, when you're ready for step 3, that the DNA gets duplicated. So if the DNA was going to be duplicated at room temperature, that meant you had to walk into a deep freezer and do steps 1 and 2, freezing your butt off, and then let it into room temperature to do step 3, and then move it back to the deep freezer, and that was kind of annoying. So what they needed was to find an enzyme in a living creature whose body temperature and normal temperature that it lived at was something like 72 degrees. Because all of the plants and animals and mushrooms that we know, they live in the same kind of temperatures we do. So a very clever person found a bacteria in the Old Faithful and other geysers of Yellowstone. And these bacteria live in the nearly boiling water in the geysers, and their normal room temperature that they're comfortable in is about 72 degrees Celsius. And you take them away from that and put them in like a regular room temperature room, and they'll freeze to death. So they took the DNA replication enzyme from the bacteria of that geyser to use in this process, so that we can be comfortable in our own labs at room temperature, and the DNA will not replicate using this enzyme until we heat it to 72 degrees. So that's kind of a cool story about how we've learned to improve the process by finding this really weird bacteria that lives in almost boiling water. So I'm going to try and play this video now that sort of has a little cartoon that demonstrates this, and hopefully the internet will be enough to make this work. So here's a cartoon showing setting the temperature to 95 degrees and the DNA strand splits. There's the Thermus aquatis bacteria enzyme. There's the primers. There's all of the loose individual letters that we mentioned. So in step 2, 55 degrees or so, the primers come in. And step 3, 72 degrees, all of the individual letters find their correct spot and snap into place. And we've duplicated our DNA. We have 2 strands instead of 1, leaving out parts of the strand that we were not interested in. So we do it again. And now we're going to watch the 2 become 4. I get a break. It's great.
We're
Danny,
not seeing the video.
Danny,
Oh, you aren't?
This is Andy McKinnon. We're not seeing the video.
Oh, I appreciate you telling me that. Let us see. I think what I can do about that is no, it's actually says it's sharing the right thing. It should be sharing it. Well, if it's not, then I'm going to just proceed because I can't see exactly what the problem would be. According to Zoom, this is the screen that's being shared. So my apologies, but if you want, I will put in the chat link where the video, so you can all watch it later. Thanks for your patience about that.
Danny, also when you lean forward, we are losing your voice.
Okay.
All right.
Thanks for that tip. Okay. How is this right here from this distance? Are we good?
Yeah, that's good.
Perfect. Okay, great. We'll continue. Okay. So now we have finished the first part and we have 1 billion copies of exactly the part of DNA that we want. But they're still too small to look at. We can't just look in the serum and see the DNA. It's still it's still way too small for a microscope to see. They're just individual molecules. So how do we actually look at it and be able to see what it is? So what we do is we repeat the process 4 more times with a few special nucleotides sprinkled in, just a few. So, for instance, we have, instead of just regular A's, we sprinkle in a couple of special A's, which I've called A prime with the little dash there. So this modified A has been dyed. It's now actually yellow. We're not just sort of calling A the yellow one. We're gonna dye this A molecule actually yellow. And what happens is, whenever that molecule snaps into place, it won't allow anything else to snap in after it. It will stop the replication right there when one of those just happens to be used, which means that some random point, as this string is going along and replicating, one of those special A primes will snap into place, and it's going to stop before it actually finishes. And similarly, we do the same thing with C and G and T, and we run the PCR again this way. So what happens after we finish this is eventually we will have many segments of every possible length. So at least once, the very first character to get duplicated will happen to be the one with the special prime that doesn't let any other characters get copied afterwards. And you'll have one tiny, tiny little piece of DNA with a single letter. You'll also have one you see under the A that's 2 long. Under the G, you'll see there's one that's 3 long, this one that's 4. You will have a strand of every possible length of all the different lengths in there somewhere. And the A batch has all of the ones that end in A. The C batch has got all of the pieces that end in C, the G batch, all that end in D, and the T, all that end in P. And more importantly, each little strand is going to glow the color of its last letter because it has a little drop of dye. Now it's just one molecule being colored, so it is not enough a dye to actually see it with the naked eye yet. And then we do the fun part. We get a piece of electrified jello. So this big gray blob represents a piece of electrified jello. It's negative on the bottom and positive on the top. And we put all of the molecules of all the different lengths at the bottom end. And all of these molecules are negatively charged. So they are going to be attracted to the positive end of the jello. And when you turn the electricity on, all of the molecules at the bottom of the screen are going to start floating up towards the top of the screen, towards the positive side of the gel. And I've already shown these molecules at the top of the screen after they've traveled. I don't have an animation showing them moving up. So what's very interesting about this is the lighter weight ones, like the one that's only a single letter, the letter C, that can move the fastest because it weighs the less. So it's going to actually travel all the way across to the other end of the gel. But the strand of DNA that's 700 long, that's the full length of your ITS sequence, weighs 700 times more, and it's actually going to move much more slowly through the gel. It's going to barely have moved at all in the same amount of time. So this is what happens. The lightest piece, just the individual C, traveled the furthest. It made it all the way to the end. The second quickest molecule is the one that's only 2 long on the left-hand side here, the C and the A. That's only 2 long, so it was able to move the second farthest, almost as far as the C. And the piece that was only 3 long traveled the third farthest, and so on. So this is pretty cool. What this means is that you can now actually see of 1 billion copies of this individual C molecule with the blue dye in the same place at the end of the gel. And there will be no other molecules with any other colors near it. It's just a big pile of 1 billion blue dye molecules. And that is bright enough to see. So you can actually see this band. This is an actual picture of what the band in the gel looks like. showing up. So you know that the first letter of your DNA sequence is C because it was a molecule from the C lane that made it the furthest. So now with millions of pieces of DNA all in the same place, enough that you see the glow from the dye, you know that the single nucleotide moved the furthest, and the only single fragment that we have is where the first is a C. So we know that our DNA sequence starts with the letter C. And the second furthest traveled was in the A column. So we know that that ends with A. So the second character of our DNA sequence is A. And we can actually read from this picture, we can read our DNA sequence. It starts with C, second is an A, then is a G, then it's a T, then it's a G again, then it's an A again, then a C, and you just sort of work your way down and you can read off your DNA sequence just like that. And if you actually dye them the 4 colors, you can put them all in the same lane and you'll actually get a colored picture like this where you can read off the DNA. Blue for C, yellow for A, green for G, and so on. And you don't have to actually sit there with a pen and paper and write it down. It's really easy to have a computer with a video camera can just take a picture of the gel and just generate that string automatically by reading what those colors are. And then ta-da, you have your DNA sequence. And it kind of works like that. So now that we know how it works, I want to talk about a bit of the challenges and sort of what you do with that data. Sometimes this is going to happen. You're going to take a look at it and you'll say, wait a minute, both a C molecule and a single G molecule move the furthest. So the very first character of our DNA strand is both a C and a G. How can that happen? Well, that's because, as I explained earlier, there's really 2 complete DNA copies inside of you, and they have some differences sometimes. And this is one of those times. The left-hand strand inside you had a C in it in that place, and the right-hand side inside, well, not inside you, inside the mushroom that you sequenced, had a G. So there's both a C and a G there. The 2 different copies of DNA had different letters. So that's something that we make a note of. And we have extra secondary codes. So for instance, on the bottom here, it says secondary codes, S equals C and G. If you see an S in a DNA sequence, that means that both a C and a G were present in the 2 different alleles, which is what we call them. And that is what that means. So there's also many copies of this region, and some of them might be different. So there's a few different reasons why you might get these double letters. So heaven help you if one strand is shorter than the other. If you take a DNA strand and you take one piece out and then you close the gap and close them together again, none of the letters will match up between the left and the right, and you will get this big blur of color on your gel and you will not be able to read it. So now we can make a tree. With the DNA sequences, we have statistical algorithms that take your sequences and they create this tree to show you how the species are most likely related to each other. And the percent that 2 species are related or that 2 individuals are related can be determined pretty well. But how they evolve that way and just how they're related, this is kind of only a best guess. There's only so much we can tell. And we kind of do it with statistics to try and figure out which picture is most likely. But you can make this little tree where you can see there is a common ancestor at the left, and then some evolved towards the top, and then they split and evolved into these other species here towards the bottom. So how much does this cost? Well, the first time that they DNA sequence, it was probably a $10 million project before they got success. But thankfully, the prices come down, and between $30 and $40 or so, if you mail a piece of your mushroom to a lab, and you only need a short sequence, like just the ITS region, which is less than 1,000 characters, you're not asking for all 50 million. That would be much more expensive. You can do it for about 30 to 40 bucks, which is pretty cool. And you can even do part of it yourself. The first half, the PCR, where you're changing the temperature all the time, you can get those chemicals and tubes and pipettes and the machine for like $1,000 and some odd dollars used, and maybe only pennies per sample for the chemicals. So you can save a few dollars, and you can do that first half yourself in your own home. You do not need to worry about touching it. You do not need to have a walk-in freezer. The second half that has that gel machine, that's a kind of a complicated machine, and that still costs many thousands of dollars, and they're pretty hard to operate. So most people will not try and do that in their own home. They'll send that out to the lab, but you'll get it half price for $15 or so. You can do a DNA sequence if you do the first half yourself, and you only need to send it into a lab for the second half. And there are some alternatives to the method that I explained that are coming up, which is pretty cool. Something called the Oxford nanopore is another method that takes the place of needing the gel and needing those machines. For about $1,500, you can buy this cool little device that will plug into the USB port of your laptop and do the second half of the sequencing for you and tell you what the sequence is. It's only 99% accurate, and you cannot tolerate any errors. So what you do is it sequences something thousands of times, and even though only 99% of them will be correct, it will figure out, having 1,000 sequences to look at, what the true correct sequence is. It takes about 2 seconds per sequence. So in 2,000 seconds, or 20, 30 minutes, it'll be able to tell you exactly what the sequence is. And after you use this machine, for a few hours, it wears out and you have to spend another $100 and get a new cell and do it again. But it's a pretty exciting new technology that's coming out, or that is out. And I know the Illinois Club is going to be playing with it this spring, sometime this month, and they'll be happy to tell us all how it works, and we'll get some firsthand news about how these new toys work. Okay. So now that we've talked about the intrinsics, about how DNA works, let's talk a little bit about the kinds of things we learned from sequencing these mushrooms that we couldn't tell by looking at them, and we couldn't even tell with a microscope. What kinds of things did we learn from DNA sequencing? So we're going to play a match game. We have 8 mushrooms. There are 4 pairs, and we have to guess which 2 mushrooms are closely related, which 4 pairs actually are most closely related. Well, if you're like me, when I was first learning, or what most people would answer, you would say that the top 2 are closely related because they're both gilled mushrooms. Both this Russula and this Agaricus have gills, so they're probably a pair of closely related mushrooms. The second that look like a pair are this coralized puffball here and this coral Romaria mushroom. They both have the same kind of coral shape, so you would be forgiven if you thought that they were closely related. For the third pair, we have a chanterelle on the left and one of the false chanterelles on the right. People are always mistaking the one on the right for a chanterelle, which is how it got its name, the false chanterelle. So obviously many people think they're the same, never mind closely related. So that is a pair that many people thought were actually closely related, since they're mistaken for each other. And finally, on the right, just like we thought gilled mushrooms were all related to each other, the spine or the teeth mushrooms, this Hericium, ball of spines and icicles on the left, the lion's mane, and on the right, the hedgehog mushroom with the little spines underneath it. Both have spines as the shape that all the spores grow on. So it was thought that they might be related. So you're not alone if that's how you would have played the game. Since the 1700s, pretty much everybody's assumed that that's what was related to each other. And it's only been in the last 20 years or so that DNA studies have shown us the astonishing truth, which is that the Russula is, as we already explained at the top of this lecture, with its round warty spores and round other cells in the background, is very different from other gilled mushrooms. It's actually pretty closely related to the lion's mane. The second pair, Agaricus, is a pretty close relative of most of the edible puffballs, are somewhat related, not that surprisingly closely, to your portobello and crimini mushrooms. The third one is the false chanterelle is very closely related to Ramaria corals. As a matter of fact, they are in the tree kind of in between two different groups of Ramarias, as if they, so it was a Ramaria coral that evolved to look like the false chanterelle and then maybe back again. Very close relationship there. And finally, if you've ever actually thought you found a chanterelle and grabbed a hedgehog and looked underneath it and saw that it had spines instead of ridges, you'll understand that they really are pretty much lookalikes of each other. And it turns out they're fairly closely related as well. And that's the true answers to the match game that things like DNA have been able to answer for us. So remember, it wasn't just DNA. When we first got the microscope, we found these clues that told us that things weren't always like they seemed. And the simple truth is that mushrooms have evolved back and forth between gills and pores and spines and gills and pores and spines all the time. It is not that uncommon a thing for the evolution of a mushroom to switch it from one to the other. And the differences that it actually takes to switch back and forth is not as big as it might first seem. If you took two gills and you pinch them together every inch or so, you would kind of be creating pores. And if you take bolete, if you take a porcini and you start tearing at the walls of those little sponge holes, you'll get these dangling spine-like fleshy things hanging down. So it's actually not much of a change for a mushroom to evolve back and forth between these three shapes. And we also have to remember that there's only so many successful shapes that will keep a mushroom competitive to be able to not go extinct. And you need to have a huge surface area to volume ratio, which means you can fit as many spores as possible in a tiny space as possible in order to be able to get your billions of spores out there to be able to have a chance to reproduce. And only a few shapes are very efficient at doing that. And gills and pores and spines really allow you to coat every edge of that gill, fill the inside of all of those tubes with spores, and coat the icicles with spores, and you are able to successfully produce a large number of spores that way. So there's a lot of pressure, evolutionarily speaking, to find one of those successful shapes to be able to do well. So finally, I want to talk about interpreting results. A lot of people think, great, I have a DNA mushroom sequence. Now I'm going to know exactly what it is, right? Well, no. Actually, the next step where you have to actually look at the sequence and figure out what the species is the hardest part yet. It's a whole university course or courses worth of material, and it is such a new topic that those courses and those books are still being written, and there's no good books explaining it yet. In the labs and the universities, the kids are kind of teaching themselves on the street in the labs because this is just also brand new. So it is very, very difficult to do. It is not something that you should feel that once you have a DNA sequence, you're just going to have your questions answered. We're not quite there yet. It's a great leap forward, but it takes a lot of careful caution to interpret the results correctly. And I'll go over a few of the reasons why. So we have on the internet a database where everybody puts their DNA sequences, GenBank, billions and billions of sequences. So you make some sequences, you put them up there, and you get to see everybody else's, and it's super cool. And there's another one called Unite that's just for ITS sequences that is a little more carefully curated to have fewer mistakes in it. And there's another great internet tool called NCBI Blast. And this is a little web program that lets you compare your DNA to all the other billions of DNA that anyone else has ever done. So you can see if yours is the same as somebody else's. Very, very cool. Or you can give it a specific set of just like 20 sequences that you hand specify. And you can say, make me a primitive tree with these 20 mushrooms in it and show me what it looks like. Really, really cool free web tools. So what happens if you find that your DNA matches somebody else's? You think that's great. I have the same DNA as somebody else. So what mushroom did they have? I have the same mushroom. Well, the sequences are up there, but very little else. When they first started doing this, they didn't realize how important it would be that everybody say where on the planet they found their mushroom. That information might not be there. So you may not even know where that mushroom that you matched with grew. You might have to call the person who uploaded it into GenBank and ask them. It may not be in there. And there's definitely no pictures. So that can be a problem. And the other thing that is really hard for people to accept is that most mushroom sequences on the internet are misidentified. They're mostly wrong. So if you see that you matched 100% with a Cortinarius violaceus of somebody else, that's probably not what their mushroom is. So if you say, great, I have a Cortinarius violaceus, you might be wrong. And heaven help you, especially with Cortinarius, where there's thousands of species and they're super hard to identify, or a Russula. Those, it's probably more like 75% of mushrooms on the internet are misidentified. So you're not going to get your answer by seeing that you matched with somebody else's mushroom. You will come closer to get an answer if you match with the one official type sequence of a species. So there's something called a type, which is the first and official mushroom. So the first one that's collected, that's the type, and that is officially that mushroom. Everybody else who thinks they got that could be wrong. But the very first one, by definition, is the real thing, and we need to sequence them all. But some of them are 100 years old, and we can't sequence them. It doesn't work. Some of them we can sequence, but we've only done it to about 1% of them. So 99% of the time, you will not find an official sequence of a mushroom to compare to. So you will kind of be at a loss of knowing which your mushroom is. More problems. You can't compare your mushroom to somebody else if you sequence different parts of the DNA. If you sequenced ITS, but they sequenced LSU, you can't compare them. You can only compare to other people who also sequenced ITS. There are at least 6 popular segments that people sequence, and yours has to be the same as theirs. Another thing, if your DNA doesn't match 100%, how different does it have to be considered a different mushroom? If there's a single difference, is it different? If there's 5 differences, is it different? How do you know when to say yours is the same and when to say it's different? Well, that's still a matter of opinion. You have to look at every DNA difference by hand, see how meaningful those differences are, and take all this into consideration. So having DNA as this tool does not mean you can stop doing all the traditional mycological work you used to do. DNA is just one more great important tool that you have to determine if 2 species are the same. But so whereas before, when looking at 2 mushrooms, we considered, do they look the same? Under the microscope, are they the same? And ecologically, do they grow in the same kind of forest? Do they like the same kind of trees? And if all those things were the same, we might say it's the same species. But if we found an ecological difference and a difference under the microscope, we might say, well, we found some differences, so maybe these are different species. It's just a matter of opinion. But now I like to think of it that DNA gives us one more tool to consider. So instead of saying, consider the macro, what it looks like through the naked eye, what it looks like under a microscope, and its ecology, consider those 3 things when deciding what your species is. Now you should consider 4 things. What you should still consider what it looks like, still consider under the scope, still consider the ecology, and consider the DNA. And now you have 4 things to consider when deciding what the mushroom is. So heaven help you, don't try to do it with DNA alone. And if you had to ask me, which of those 4 is the most important? Well, we've already learned that what it looks like to the naked eye is not very important. So pretty much everybody knows that that's not the most important thing to consider when determining what a mushroom species is. So is it DNA? No. I think ecology is probably the most important thing. There's so many lessons where how the mushroom makes its living, whether it's mycorrhizal, living on tree roots, or whether it just digests dead matter, is such a huge difference. That is a very meaningful difference. And there's no way two mushrooms can be very closely related and have two very different ecologies like that. You can have two mushrooms with the exact same ITS DNA sequence with different ecology, and they're still being considered different species. And you can have mushrooms with different DNA and the same ecology, and they might be considered the same species. So ecology is probably the #1 most important thing to consider, and DNA would be second. And macroscopic, what it looks like to the naked eye, dead last. Conclusions. Was it all worth it? Well, I can't help but think about all these scientists that spend their entire lives trying to classify mushrooms. The famous taxonomists of the world, Fries, Smith here in America, Peck in New York back in around the year 1900. They devoted their entire lives trying to figure out which mushrooms were actually closely related to each other and belonged in the same genus and which didn't. And they never even knew for sure that they'd ever find the answer. It was kind of thought that it was just a matter of opinion. People disagreed all the time. There were people who said, no, that gill polypore is more of a polypore than a gill mushroom because it's hard and tough and growing on a tree. And there were people who said, no, it's got gills. There's no way you could just evolve and change from pores to gills. It's really a gill mushroom that just kind of happens to grow on trees and be tough. And matter of fact, in their monograph on the North American species of Hygrophorus, Hessler and Smith wrote, "This matter will probably continue to remain one of opinion, as it has in the past. The end is not yet in sight." Well, thanks to having this extra tool of DNA, the end is now in sight. I mean, it is still and will always be a matter of opinion what the differences in DNA mean. Whether or not you should think of two things as the same species or a different species, that may always be a matter of opinion. But pretty soon, we will really know exactly how genetically related two mushrooms are to each other, which will have great meaning in finding other deadly poisonous mushrooms, finding other mushrooms with the same ecology. We'll actually have those kinds of answers. And it's been really fascinating to find out when these taxonomists were correct in their guesses and where they were wrong. And I really wish I could go back and tell them about their life's work, that we have some answers now, and just how happy they would be to see the advances of how much more there is to learn because of this important tool. And I wish I could go back and tell them. So thank you very much. That is my talk.
Danny, thank you so very, very much. What
You're very
a
welcome.
super presentation. What a
Excellent.
great presenter and a great teacher above all.
Thank you. And thank you to Kim, who posted the link to the video. So everybody can now go and watch that now.
All right.
Just more congratulations on the wonderful presentation that Danny provided.
Thank you.
Are there any questions from the audience?
Yeah, so basically a lot of lovely comments of how they enjoyed it. Enlightening and brilliant. Someone mentioned something about a microcosm.jgi.doe. Have you heard of anything as such? They're wondering if it's a useful tool.
I don't know about it.
Yeah.
Mel, can you give that again, please?
I will type it into
The chat.
the chat.
In the meantime, Alan is asking the question, can you clarify the mushrooms regarding ITS, all species have junk?
Yes, it is a very particular junk region that is identified by 20 characters so that the beginning of this junk character has these 20 characters at the beginning, so you can find it. All other types of junk regions, including all of yours, have different set of 20 characters at the beginning of them.
Does that answer your question, Alan?
Alan, you can press the space bar and ask the question yourself if you want to clarify. No?
I also note that Adolf pointed out that there are 23 pairs of chromosomes, which is really 46, and that is correct. That is correct. It's actually 23 pairs for 46 chromosomes.
And
Right.
another complication, some basidiomycetes have 2 nuclei.
I think I got it.
I unmuted. We got it. If you wish.
I think
to
we
speak.
got it.
Yeah.
Yeah.
Okay. I muted you.
Alan,
Yes, sir.
we can hear you talking. Do you want to
Yes.
ask a question?
Yes, George had a question, but I think we had it answered. Thank you.
All right, great. Can you mute yourself again, please?
Okay, there's a question from Andy, more or less everybody else's. How local mycological societies adopting DNA technology? I guess it's the difficulty of getting the expertise and / the equipment.
So I've kind of volunteered to be a person to look at any DNA sequence from the Pacific Northwest to figure out what it is. And I'm working on making tutorials and videos to help show other people how it's done. At Key Council, I'll be the speaker talking on this topic as well, and going into more depth about how we do that. The Bellingham Club has their own PCR machine. They're doing step 1 of their own sequencing, so that they only have to pay for step 2 to send it to a lab. It's getting cheaper and cheaper all the time. So it's really a thing about education. Fundus, which formerly known as Mycoflora, they have been helping clubs get sequencing, and they have donated mycology time of some of their mycologists have interpreted the results, and they are making tutorial videos and slide decks to show people how to look at the DNA and how to interpret the results to help train more people to do this. And they're putting those up on the Fundus website right now.
Yeah. Oxford Nanospore, I've been in touch with them for the past 18 months, and they are willing to do workshops.
Cool.
Yeah, I did. To follow up on my question, Danny,
Yes.
I'm wondering what your advice to the South Vancouver Island Mycological Society would be in terms of developing expertise within our society.
Well, I am pasting a link. That link is to a website where I am trying to compile, this is probably a decade-long project, but for every mushroom species, for all 4,000 mushroom species in the Pacific Northwest, which you can use MycoMatch to find out what they are, put the DNA for each of them up there, and to say whether or not we have the DNA yet, or if we need it. So you can collect some rare things at a 4A, go up to this website, find out if it's been sequenced yet in the Pacific Northwest. If not, save it in sequence and send it to me, and I'll post it on the website, and we will have a Pacific Northwest comprehensive site where all of the DNA sequences that anyone can download and see are, and we'll have a list of everything that we still need.
Does that answer your question, Andy?
It does indeed. Excellent.
You're going to volunteer, are you?
Excellent, Danny, and thanks so much for the presentation tonight.
Yeah.
Lucy.
Pleasure.
Mel,
Yes.
we have a committee at the moment, which Mel is a part of from the board. Mel, could you just say briefly what we are doing? We're looking at the whole problem, or not a problem, adventure.
Yeah, so Andy and Shannon and Kim and Joey from the club, as well as Jason and Ian Gibson, they're all a part of it, and we all have different tasks to try and figure out if the club can afford to begin sequencing things, or just to help log away information from certain specific finds. So it's something in the works. I still have the PDF that I need to send to the board to approve. There's a lot that's involved, especially because we're a nonprofit, and in order to get things sequenced, we obviously have to pay for it. So the problem is trying to figure out how to get that properly done. And then, of course, what's most important, is it to get things sequenced, or just to dry things and prepare them and to find out their ecology and all these other things, right? So there's a lot more to it, and there's a team that's taking care of all of that. So yeah, that's kind of what we're having in the works specifically for our club. But I mean, for every club, it's very different, obviously, depending on who's volunteering and who's a part of it as a mycologist or a scientist. So yeah, it's complicated.
We're talking about documentation, vouchering, herbariums, and then sequencing.
Yeah.
Having
So
it
there's
as a comprehensive approach to the whole subject.
To make sure to provide consistency, right? That's kind of the issue that I find I'm hearing, especially from you, Danny, as well, is consistency. So that's kind of what we're trying to do, and it's still in the works. But anyways, I have a lot of questions for you, Danny, on Facebook. So bear with me. I'm going to scroll through these, and patiently we're going to go through this.
I had a question. I had my hand up here, and have been answered here. Danny, have you done anything with the barcode of life?
I've heard of it. So the barcode region is the name of the ITS region. That's what everybody has decided will be the first thing to sequence. If you don't know what region to use and you want to kind of do what everybody is doing to contribute to the barcode of life project, you'd sequence ITS. So that is the region that I also always work with, because it's what's mostly available. And
so it's a bit more than that, because there's essentially, they're doing more than that. They're coming up with particular sequences that are for a wide variety of organisms, insects, and other stuff like that. But the one thing that they do have is they actually have a repository to put all the metadata, pictures, and all that go along with the specimen that you've done the DNA on, which is a point that you were indicating there's a difficulty there. And we actually had, for a couple of forays, we actually had all the metadata required, pictures, mushrooms ID, and they actually did a couple hundred DNA sequences for us, and have that. And there's actually a project that's on there that came from a few years ago. This was 5 or 6 years ago, before David's time. I was involved in that one. Shannon was involved. Nothing's happened with it there, but that's something that we did, and they basically have taken that task to be able to put all that stuff data together. So I strongly suggest you may want to take a look at all the work that they've done there. There's a huge amount of work to be able to get that metadata together, all the photos, all the specimens that were in there were sessioned at DAVFP, and that's where those samples were put there for that particular material was collected.
Sorry, Tony, mentioning, when you say you, does that mean the committees that are just kind of conferring at the moment? Or Danny?
Well, I was talking to Danny specifically.
Okay.
I said that maybe not there. I did bring this up earlier when David brought it up, and I sent a suggestion out there, but I don't know, the email was ignored as far as it goes. Shannon knows about it there. Nothing's been done on that particular piece of it there. So I can share with the committee for SVIMS on what's been done on there. I know Shannon looked at some of the sequences there and some of the tentative IDs that came up from doing the blast, and they sort of said, well, we don't believe that's what it is. So there's something that was going on in that particular case, but some of them were okay. So we've got a couple hundred specimens that were put up there on the Barcode of Life iBold site.
We'll bear that in mind, Tony, and if you don't mind providing some of that information to the subcommittee, that would be much appreciated.
Of course.
Right? And people, if you want to talk, just press your space bar and we'll try to
Adolf
accommodate
has a
you.
question.
I have to say that Oluna has about something like 600 or 700 collections sequenced, and it produced quite a few new species, but she wouldn't do the sequencing by herself, in spite of the fact that she is a very skilled lab person. So what she does is that she sends the collections to the specialists like Cortinarius to Joe Amirati or Hebeloma to Henry Becker, and leave the work on them, because as you could see, there are many steps when you can go wrong when you are doing your own sequencing.
And her collections are especially valuable because of all the great collection data and microscopy and everything that's been worked up on them. They're very valuable collections.
There's another few more questions. Liz, are you able to ask Danny your question?
I would say start with taking a notebook to the field.
Liz, are you still there?
No.
No.
Mel, you have a number of questions coming
I do
through.
have a number of questions. Let's get to some of these because we'll get
Okay,
through all
well,
of them.
yeah, I'll skip to Ken then. Oh,
What may I just ask a couple to kind of get them out of the way? They've been
Sure.
waiting for some time. What program do you use to edit your sequences?
Mega is a free program that can edit sequences. And so since it's free and it does a pretty good job, it makes much better trees than that Blast website can do. Part of the tutorials that I'll have will be covering specifically how to do things with Mega.
Okay.
There's something called Sequencer, which a lot of the pros use, but they won't even tell you how much it will cost until you call them and talk to a salesperson. So think thousands of dollars a month.
Yeah. So next question. Oh gosh. Can we categorize fungi into an advanced group or primitive group based on DNA sequencing?
Well, we do have things called basal clades, which are kind of like the most sort of primitive early ones. It's not quite right to think of them as these evolved first and then these evolved later and they're more modern, because all of these species might have only evolved recently, but it would be true to say that we do find the sort of basal areas where there's a more direct connection to the very, very old species. And we can see sort of the way things evolved, how gills came kind of late in the game, how jelly mushrooms were one of the crusts that just simply grow on the wood flat, and jelly textured mushrooms were some of the first things to be evolved. And then came pores and then came gills. And so we do kind of get a picture of that, although it's not quite accurate to say that these are the older species and these are a newer species.
Interesting. Great. Another question quickly. Are there any PCR machines you recommend for people who are just starting off at home sequencing?
Oh gosh. We should check with the Bellingham Club because they just found a used one of whatever it was, and Sharon's been using that, and so they'll have a lot of feedback on it. I haven't used a lot of different ones personally to be able to recommend one over the other, but I know it's pretty cost effective.
The advantage of Oxford Nanopore is that they do the long sequencing, which can be a benefit. At least in medicine, we are using more and more the long sequencing. And it's revolutionized medicine, at least cancer medicine, and a lot of the other subspecialties. It's had a major consequence. In fact, one guy described a young punk to an old guy like myself, that you were in the dark ages 5 years ago. It was very glad to know, very pleasing to know that I was in the Dark Ages only 5 years ago.
There's another question from Cam. Could you tell us a bit about the Untitled DNA site and what you were doing with it?
Yeah, you basically, I can quickly share my screen here. Let's see, where is it? Right here. So let's see, share. There. Okay.
Yeah.
So you go to this website here, and I have basically a tree of life that shows all of the different families and genera. So we can sort of collapse it. We can get a sort of a better idea of it. So here's all the Basidios in black. Here's all the Asco orders and families in red. And you can sort of open up the Agaricales and you say, here's most of the colored spored mushrooms are all in one place, which is kind of cool. And let's take a look at, you have these really primitive guild mushrooms where they were mostly clubs and corals and there was only one guild mushroom. They have a family that had a bunch of cups and only one guild mushroom. You have the ones that we just don't know yet where they belong because we haven't figured it out. And then you have the major family, the sort of waxy cap family, the Marasmius family, the Tricoloma family. Then you have the Amanita and Pluteus family, and then you have all the brown sport families. So you can sort of take a look at the tree and then pick your favorite thing, like Stropharia. Go to the Stropharia page and click on Laradiomyces, and you'll see pictures of all the ones that we've got sequenced, where we can actually say what I know about what the DNA has told us. And I can say things like, we need local collections with a ring because we still haven't seen if that's really a different species or not. And you can sort of see what we know. And only a few of those are clickable yet. A lot at the top are. Then as you get in here, you see most of these families are not clickable because I haven't done them. And when you get past the, to all of these orders and anything in ASCOs I haven't done. I've spent maybe 1,000 hours on it, and there's still probably a few thousand hours to go before it'll be complete.
Does that
That's
help
incredible, Danny.
Yeah.
Does that help your question, Ken?
Kim?
All right, I have a couple more questions if that seems like it may have answered. Someone else asked, they understand that some species exhibit both mycorrhizal or saprophytic ecologies at different times. How do you recommend addressing this complication when using ecology as a tool for identification?
That's a good question. I don't know, and there's a lot we don't know about that. I would imagine that the ability to form mycorrhizal is a very interesting idea. So I would sort of consider anything that has the ability to do that. That's an interesting part of its ecology. Yeah. But there's so many things that we just don't know. That's why it's really fun to work in mycology, because there's so many unanswered questions you can help answer. Yeah, that's the flip side to not knowing very much. There's a lot of opportunity to learn new things. Yeah.
Or theories. How far off in time will a comprehensive library or database become available to help identify species? We're talking about how many millions of fungi that still need to be.
Right. Well, I hope to have a database for all the Pacific Northwest in about 10 years. Yeah. But I mean, it's happening quick, I think. This is going to, snowball, or mushroom would be probably a better analogy. And, things are, it's getting cheaper and cheaper. Fundus has been trying for years to get sequencing down from 30 to 40 bucks to like 10 or 15 bucks. Because they think like one more halving in price and it's going to be really explode. People are going to say, okay, 10 bucks. Yeah, I'll pitch in to do that. It's really going to, it's really going to help things come along as soon as we just get one more halving of the price.
Yeah. Yep. Gosh, there are a lot of questions. Hang on.
There's one more from Heather while you're looking for.
Okay,
What do you recommend for preservation to send off mushroom samples for DNA sequencing? Is DNA
Dehydrate them.
Pardon?
Dehydrate. I wouldn't freeze dry, because then who knows what's going to happen as it thaws out. Put them straight into the dehydrator before they start molding, and for longer than you think, for 24 hours, slice thin until they're absolutely crispy and have no bend and give to them whatsoever. The better they're dried, the better they'll last. They're people who are really good at DNA sequencing can get DNA out of things that are 100 years old if they're preserved properly, and that is dry them to a crisp. Like say 110 degrees, so you don't like cook them. So it's a very moderate temperature for like an entire day and sliced thin.
Okay.
No hotter than what degree, let's say?
110 or so.
Yeah.
I mean, nobody knows for sure, but some people worry that too hot could cause some damage that wouldn't allow them to last 100 years. We'll have to wait 100 years to see if that's true or not, but it seems to be, people feel pretty confident that, drying at 110 is not going to hurt them, and just let it go for a whole day. And that's a pretty safe way of doing it.
Okay.
Thank you, Danny. And Kurt and
Paul
Melanie.
Kruger is trying to ask a question. Paul,
Fahrenheit or Celsius?
Oh, that is Fahrenheit. My apologies. I've been corrupted by the time I've spent in America.
I love that.
Yes. So that was a Fahrenheit temperature. I apologize. And at least in my presentation, I remember to use Celsius. Yeah.
Olona does it at 55 Celsius.
Yeah.
That sounds good. So someone's about to start taking soil samples from a year-old burn site in an effort to determine the rate of mycorrhizal recolonization in the burn area. Would he want to compare the eDNA data to which genetic bank best suited for this?
GenBank's the easiest and the quickest. And even if you don't know for sure what your species is, you'll see a whole bunch of matches in the same genus. You'll probably get to genus, and that's what they want to know, what kinds of things are on there. Use the BLAST website to blast against GenBank. It's the largest one, and it just takes a matter of seconds. Something that, when I find that the free web program doesn't quite work as good, and I use the mega program, which will give better results, it will give better results. It will take 100 times longer for the computer to crunch it out. So you only want to do that if you have to. There are supercomputers on the other end of the NCBI website that you just put your sequence in, and what would take your computer 24 hours, in 3 seconds, it gives you the answer. It's just great.
Wow, that's incredible. Someone from Australia, Teresa, says, thank you so much for this. She doesn't have words to express her delight and how simple it was to follow. It was brilliant.
Oh, that's great to hear. Thank you.
Yeah.
I really appreciate that.
There were some people from across the country here, and across the globe apparently, so that's fantastic. There's another question. Is it possible that the same kind of mushroom would grow in a different habitat? Say the same chanterelle would grow under deciduous trees versus coniferous trees?
Yes, different mushrooms have different levels of fussiness. So there are some that will only grow under one particular kind of pine tree. And there are some that will grow under any kind of pine tree. There are some that will grow under any conifer tree, and there's some that will grow on conifer and deciduous, and they don't care. So they all have different levels of how fussy they are about what their ecology is. But in general, there's still important things to know about, to learn about the ecology that can teach you a lot about the mushroom more than any of the other aspects can.
Awesome. Thank you. I believe that's it from my end. So is there anyone else?
There's one more question from Kim. Do you find the unite species hypothesis useful?
Yes, I do. I will be talking about that in my tutorials when they exist. And because unite can draw a map. So when you take your DNA, it'll show you a map with dots of where DNA that was at least, say, 99.5% the same as yours, where it was all found, and draw it on a map. You don't have to read the text, and spend hours looking for the word Germany or anything like that. So Unite is a great, valuable tool as well. It's definitely an important piece of all of this.
Thank you, Danny. I'm going to have to terminate the meeting. We're almost coming to the end of our Zoom time. I gave 3
I
hours.
think Paul had a question, Paul Kroger.
Paul?
Hi, I was just wondering, is there a way to search? I keep getting SH species hypotheses numbers attached to my collections. Can I search that number under access Unite and somehow find out?
Oh, with that SH number, I think so, yes. I'll figure that out and I'll email you later tonight.
Thank you very much.
Yeah.
Well, thank you, Danny. That's
bet.
fantastic. I'll be in touch with you tomorrow.
Great.
Great.
It's a lot of fun. Thank you. It's really
Fabulous.
great to see everybody too. This is really cool.
Have a great night. We'll try to get all these references and all, and I'll bug Danny.
You
All
too.
right.
Okay.
He knows I'll bug him. I've got his number. Thank you, Danny. Thank
Thank
you.
you.
Thank you, everybody, for attending. Have a great night.
Yeah, you too. Thanks.